We have quite a few questions around LNPs. We had several questions in the registration process and some coming across right now. So let's shift gears and talk about LNPs a little bit. Starting at a high level, what LNP variables impact immunogenicity and stability? Christian, start with you.
Yeah, I think most LNPs that are used are composed of similar components. You have a cationic component inside that complexes with the or interacts directly with the mRNA, and you have helper lipids, PEG lipids, and I think most of the both marketed mRNA drugs apply to this concept, and I think a lot of LNPs in the field do as well. I think one big topic with regards to LNP nucleic acid drugs is it's a very complex system, and you have this LNP suspension that has this limitation with regards to suspension stability. So a lot of formulations tend to aggregate if you treat them wrong, especially during manufacturing processes, meaning that single LNP components stick together and agglomerate, or the other option is that you have a loss of integrity as the mRNA is released. Of course, that's an undesired effect if you lose the integrity of the LNP, because then the mRNA is not active anymore. But I think the bigger topic with regards to immunogenicity is the agglomeration of LNPs to larger fragments, which then have immunological effects that are, I think, difficult to be controlled. If this can be excluded and you can stabilize this suspension, that significantly improves the safety profile, I would state, to LNPs. Apart from that, of course, it's important to choose the right components, so the right LNP components, right cationic lipid, right helper lipids, and proper shielding with the PEG shields to avoid undesired side effects of these LNPs.
Patrick, anything to add there?
Yeah, principally, currently we have maybe a glimpse on some aspects that might play a role in immunogenicity and reactogenicity. On others, we do not have a complete understanding. So the current systems are usually really at between maybe 60 to 100 nanometers in size. The data on if size is a main contributor on the immune stimulation is inconclusive so far, at least from the literature that's available. So, and it's also not easy to study, because sometimes it's an issue to, within certain composition, to generate then a lot of different lipid nanoparticle sizes in a 10 nanometer steps, and then test them. So usually if you change one parameter, also other parameters change. That makes it quite complex. I think the understanding is growing, and we have a lot of people working on different aspects, and we will see in the future more and more data coming out, also mode of action data that is really looking into specifics. We will have a lot of human data coming in with the rise of more and more systems going to the clinical phases, and that will increase our understanding. But at this point in time, it's not like this, that you can design your lipid nanoparticle and from the drawing board say that will be immune stimulating or that will be immune silent.